Welcome to the next lecture of this course Environmental Biotechnology, and in this course we are going to complete our discussion on the methods in microbial ecology which are used in relevance to environmental biotechnology. In today's lecture we are going to highlight a particular topic which is the metagenomic approaches, and this basically comes under the culture independent analysis methods that we have been discussing for some time now. Now, what is metagenomics? The word meta refers to transcendent, that is something which is beyond or above the range of normal or physical human experience, something which is large and something which is really able to provide some comprehensive information or understanding on a particular topic or a particular subject. So, now with respect to this community analysis, microbial community analysis, the use of the term meta along with the genomics refers to some kind of protocol, some kind of understanding, some kind of advancement that the scientists have made utilizing the concept of microbial genomes. That means, it is kind of an all inclusive concept, that genome means collection of all the genes present in a particular environment. And so, the metagenomics basically has emerged as both research field as well as a research technique. So, as a research field or a research technique, we can see that it has actually diversified, as I mentioned, into two different directions or two different components. In one of the components, that is basically targeting the methods and approaches, it helps us to circumvent the unculturability and the genomic diversity. Now, you are already aware of the unculturability aspect of any environmental community, and the concept of the genomic diversity has emerged lately, and what we have observed that within the genomes of individual species there are enormous amount of diversity. For example, if we are using 16S ribosomal RNA gene, or we are isolating a couple of a definite or a specific group of bacteria and trying to look into the whole genome sequence of those isolated bacteria, we are able to find out that within the members of the same genus or same species, for example, within E. coli, there may be different strains of E. coli. Now, if we look at the 16S ribosomal RNA gene, these E. coli strains might all appear same, they might not also, sometimes they may show some variation at maybe 99% similarity or identity level, but many a times we will be able to find out that all the strains of E. coli are E. coli by virtue of their 16S ribosomal RNA gene. But if we look at their genomes, all the E. coli strains that we have with us, if we look at their genomes, that means the complete genome sequence, we will be able to find out that the genomes are significantly different. How significant this difference could be? This could be like 20% up to or more than 20% genes are different from one E. coli to the other E. coli, but all are E. coli. What that means to us who are working on environmental biotechnology? It means a lot to us, because suppose we identify there are certain E. coli or there are certain Vibrio or there are certain Pseudomonas in our samples, we cannot rely only on their taxonomic information, that since there are Pseudomonas or there are Vibrio, so they might be pathogenic. So, we have to take care about the quality of the treatment, etc. But it may appears that the genomes of those Vibrio are having significant dissimilarity with genomes of the pathogenic Vibrio, and essentially we may not implement such kind of treatment processes if we even have those organisms. It is more appropriate when particularly we work on certain specific organism based bio remediation, bioenergy, or sequestration of carbon dioxide, or removal of phosphate, or removal of nutrients, etc. We cannot just rely on the taxonomic identity of one organism. So, that means we need to have some technique which will go beyond 16S ribosomal RNA gene and will help us to take into account this genomic diversity, that is the diversity that is there within the genomes of even the same species or same strain of the particular species. So, now in this respect, this new technique that is called the metagenomics helps us to overcome surely the unculturability, because it is a cultivation independent method. That means, we work on the extracted nucleic acid directly, but it also helps us to address the issues of genomic diversity. That means, if I want to know that how many different types of E. coli are there in my sample, although they may be similar or identical with respect to their 16S rRNA gene, still I can find out that. Now, this particular aspect, these two aspects, the unculturability and genomic diversity, these two were considered to be largest roadblocks to advances in the clinical and environmental microbiology, because all our processes, all our diagnostic and treatment systems, our ability to control the process, etc., were relied for a long period of time on certain type strains or reference organisms. We were unable to know that actually what are the other possibilities with respect to that particular strain, but having a different genomic abilities, and the genomic ability is surely connected to their metabolic functions. So, the metabolic functions remain uncharted and also unexplored, since the genomic diversity and the genomes were unexplored. Now, it is also true that we cannot isolate all the species of E. coli, for example, or all the species of Burkholderia or all the species of Vibrio, but is there any way that we can still analyze the genomes of all those Vibrio, all those E. coli, all those Pseudomonas, whatever may be the genus or strain in my sample, so that I can take appropriate decision that this sample has this kind of genomic diversity, genomic potential. So, this should be utilized or this should be handled in an appropriate way. So, that those roadblocks, unculturability roadblocks and genomic diversity roadblocks, were actually at least significantly removed. Now, it also, this metagenomics as a method or as approach recognizes the need to develop computational methods, because we are going to rely on nucleotide sequence data, and it is obviously not one or two sequence reads that any individual can handle manually. So, it is going to be huge, and when we try to align the reads, one or two reads aligning individually or manually is possible, few tens are also possible, but when we think of few lakhs or few millions reads, it is not possible. So, we are developing computational methods, so or already developed a number of computational methods in order to analyze these data. Data is basically here the nucleotide or DNA reads that we generate during the next generation sequencing technique. So, that actually the development of the computational methods, that helps actually to understand improve our understanding of the genetic composition of the activities of the communities, and they are so complex that they can only be sampled, never completely characterized, because we cannot have the absolute value that what is the 100% of that community actually. We use different methods like 16S methods, like earlier we used to have the culturable methods. So, we are only deriving some information based on our capability, but these computational methods are enabling us to achieve to some extent close to the absolute number, absolute parameters, those are there. So, in the second sense, so that first sense was the method and the approaches, and the second sense, the metagenomics is basically in a research field. It means that this new science seeks to understand biology at the aggregate level. So, it is something which is, maybe I can use a corollary term which is called systems level understanding. So, it is not about one species, one organism, one particular gene, something beyond that. And by this time you must have understood that in real environment microorganisms are not alone, they are so many. They alone, the prokaryotic microorganisms like bacteria and archaea, they alone interact, and they all the time they perform within the guilds. So, they form metabolic links among themselves. So, they are all the time interacting with themselves. So, we cannot dissect one by one and identify a few and claim that we have understood certain wastewater treatment process or certain biogas plant system or something like that. So, we need to have processes by which we can actually transcend the individual organism to focus on the genes in the community. So, instead of identifying one or catch hold of one or two bacterial genus or species, we need to look into the genes which are there in the community. No matter the genes belong to whom, if the genes are there, the possibility is high that the genes are going to function. And how these genes might influence each other's activities in serving collective function, that would also require that we have some methods through which not only all the genes and their regulatory modules, like when we look at the open reading frame, we can identify their regulatory modules, but also how these genes might be controlled in terms of the different environmental factors, changing conditions. It may be the changing conditions in a wastewater treatment plant, it may be the changing because countries like India, we have so much of climate change, like the heavy monsoon, sometimes it is a dry spell. So, the quality of our wastewater, the quality of the river or the lake water, because of this kind of natural fluctuations, they are changing all the time. So, how our drinking water treatment plants will take care if we are able to comprehend that these are the factors which actually control the microorganisms which are present in the particular water. It may be drinking water or it may be some other type of systems. So, now so long the NGS based methodological approaches that we have been studying, it has basically some components which are very clear to us, that from the sample we need to get the genetic soup, that is the mixture of all the organisms DNA and RNA, and then perhaps in earlier experiments or earlier lectures we have discussed that a PCR of the target gene used to be a very important event, because the target gene was a kind of a key component to characterize the genetic soup or characterize the community. Suppose it is a wastewater treatment plant or a kind of a activated sludge or a microbially enhanced phosphorus recovery plant. So, we were all the time focusing on a set of organisms that we knew these are the organisms who are responsible for this process. So, a set of specific primers, often it is the 16S ribosomal RNA gene or some other gene of interest, the functional genes, and essentially we were analyzing the data in order to answer the major questions, which are like what microorganisms are there, what are their potential, how are they interact, how are they responding to the external perturbations, and how can we make use of these organisms or this knowledge that what are the organisms present in our environmental biotechnology process. But is it enough? During our earlier lectures, when we discussed about even the next generation sequencing based amplicon approaches, where 16S ribosomal RNA gene or similar genes are used to generate high number of reads, maybe millions of reads, and then analyze it, we have identified that there are some specific limitations. The most prominent limitation is it is a PCR based process. So, it is using PCR primers, which we know that they are well optimized, they are very standard primers, but who knows, the target gene might have some mutations even in the PCR primer binding site, and even if the gene is there, the gene may not be amplifying, because the gene is having some new version, some modified version of the nucleotide sequence at the PCR primer binding site. So, even if the gene is there, so I can actually clear one particular water as saying there is no Vibrio or no toxic compound producing strain or no pathogenic strain, but actually there might be some, because the PCR primer was unable to identify bind that particular gene because of some mutation or some alteration, and also the availability of the particular template DNA and all other issues were there. So, we discussed earlier all these things. So, basically the metagenomics when it evolves first, it was maybe around 25 years ago or so, and the last 2 decades, over the 2 decades, initially it was basically the fragmentation of the DNA, of the total DNA without any PCR, and the fragmented DNA pieces used to be cloned, and each of the clones were screened for their sequence through Sanger sequencing or tested for their function. Now, the advent of the next generation sequencing technology has actually provided easy access to the separated approach. So, it is now not reliant on the sequencing of PCR amplicons, number one, like we do not need to PCR anything, neither we need to wait for the time and the money that large number of clones are to be sequenced, because initially we were actually using, or the scientists were using Sanger sequencing method. But the NGS method is capable of providing the information in a kind of a large scale, which is basically metagenomics. So, now if I go ahead with this, that we have the genetic soup that we discussed earlier. Now, earlier if you remember, we used to do a PCR reaction with this, but now no PCR reaction. The genetic soup is taken, that is the extracted DNA, or it may be RNA, that I will come up sometime. So, total nucleic acid of the DNA is straightaway fragmented using some methods which are chemical method or ultrasonication method, and it is fragmented into small pieces. Now, each of these small pieces were subjected to next generation sequencing methods. So, that is the beauty of the next generation sequencing methods, that these methods are developed in such a way that they can handle a large number of, truly large number of short reads which are generated, and then larger number of reads are produced. The reads are produced after the sequencing is done. So, we get the nucleotide sequences which may be millions and sometimes even billions of reads. So, if we wish, we can have billions of reads. So, what are these reads? These reads are basically produced out after the sequencing of the sample. Now, what was the sample? Sample was the short fragmented DNA. No PCR, it is directly the DNA was fragmented, and the small fragments, obviously there will be some kind of a size selection process, because the sequencing technology would prefer to have reads or the template DNA of almost similar size. So, we will take enough starting DNA, so that even if we have some smaller fragments, we may need to sacrifice them, but we will still have representative DNA species, and eventually we will get the reads. Those reads are going to be processed through the analysis platform. Now, these reads, now unlike the 16S ribosomal RNA gene or 18S ribosomal RNA gene or any kind of functional gene analysis that we discussed earlier, these reads are not for any particular gene, rather they are for all the genes those were there in the sample, or those are there in the sample. So, now when we try to analyze these reads, we will get the information that will be entirely at the different level. So, that will obviously answer our questions, but with less bias, because PCR is not incorporated in this experiment or in this protocol. Now, instead of amplifying the phylogenetic or functional markers from a sample of extracted DNA, the DNA comprising genomes from all species present is shared into a smaller fragments and sequenced directly. So, this is basically having the intrinsic step that we extract the total DNA. Sometimes in some cases, suppose for groundwater or such sample, we can actually have the cells sieved out first, and then because we may have to use a large volume of water, or particularly water, maybe thousands of liters of water, maybe because the microbial load may be less. So, we may not have sufficient DNA at the end for the sequencing purposes. So, we may need to pass a large like 5 liter 10 liter water through the filter, which are with very minute pore size 0.22 micron or even less than that. So, that the cells are collected, and then the cells are lysed. No bacterial cells are isolated, but they are separated out in case of water particularly, but in case of solid sample like rock, sediment, etc., or sludge kind of samples, we generally do not need to extract the cells. But Again, some scientists or some groups or some researcher might prefer to enrich and get their organisms isolated separately as a bulk mixed culture. So, that they can do, but ideally the protocol may not require that. Now, simply as I was mentioning, the DNA is fragmented, fragmented DNA is subjected to sequencing. So, following sequencing we need to go for the assembly. Assembly means the reads are joined together, the short reads which are generated are assembled. So, for the assembly there are certain logics used. The assembly is done in such a way so that the pieces which perhaps belong to the same organism are joined together, and the intact contig, that is the contiguous sequence read, is generated. Now, once we have this read which are contiguous, as you can see the reads are assembled to have these larger pieces of DNA, they are subjected to phylogenetic binning. That means their phylogenetic identity, taxonomic identity is derived, and based on the taxonomic identity we bin them. So, binning is optional, some people like to bin it so that you can identify how many organisms are. So, if you are doing a binning process, then possibly you will be able to reconstruct the genomes out of the metagenomes, but if you are do not doing binning, then still these contigs may be used for identifying the genes present there, that is called the annotation. Now, the metagenome assembly that I was referring to. So, assembly is the process of combining the sequence reads that is generated by the sequencer into contiguous stretch of DNA, and these are called contigs. So, very short reads like 200 to 300 nucleotide reads generated are joined together. Now, this joining is done by the bioinformatic tools, which is based on certain assumptions. So, that the only the reads which are generated from identical type of genomes or the organisms basically. So, they are going to be joined together to establish the or to get the contig. Now, assembly merges the metagenomic reads, metagenome derived reads from the same genome into a single contiguous sequence. Now, the same genome is not visible to any bioinformatician or not even for the computer, but there are certain logics which are used to anticipate that these reads might be coming from the same genome. It is basically some signature sequences or certain other residues which are identified, and based on the bioinformatic program, because there are multiple programs, multiple pipelines are developed based on the type of samples, there is some kind of requirement, and that is useful for generating the longer sequences, which can simplify by the bioinformatic analysis relative to unassembled short metagenomic reads. So, it is always preferred that the short reads are assembled into contigs, and then the contigs are joined together to make larger fragments. If you are getting a good quality data and able to use a proper bioinformatic pipelines, perhaps you will be able to join the pieces like the contigs together to reassemble some genomes, which will be called metagenome assembled genome or MAG. Okay. So, then you can actually characterize the entire genome of that MAG, which is a kind of a reconstructed genome, but from that you can actually develop further procedure and then verify them. So, this is some graphic is here that we first go for the contig assembly, and then super contigs or genome assemblies are prepared, and if you are having a very good data and you are using a appropriate bioinformatic pipeline, then the shorter pieces joined into medium large pieces, and the medium large pieces are joined to larger pieces, and the larger pieces are joined to have the almost entirely complete genome. Sometimes the genome may be incomplete, because you may still identify there are certain gaps and some contamination may also be there, but this is possible, and it is a kind of a regular event for scientists who are using metagenome assembly technique to get the genomes out of the metagenome. Now, in some instances the complete or nearly complete genomes can be assembled, which provides insight into the genomic composition of uncultured organism, because otherwise these organisms are not possible to be cultured. So, we do not get their genomic sequence with us. So, it is only the metagenome analysis which help us to get the genome even if the organism is uncultured. So, here again there are many methods through which this can be compared. So, here for example, the SPADES and MetaVelpet and IDBA, these are different assembly pipeline, binning pipeline, these are the softwares or the bioinformatic validation pipeline, etc. They are developed and used to have the genomes, or even you have the contigs also. So, these contigs itself are good enough because you with these contigs you can still go for your annotation, that is identifying the genes that is there, and then identify the organisms which are present, identify the function that they can carry out. So, what is the major outcome of the metagenomic analysis? So, basically the metagenome is still able to answer the 4 fundamental question, that is who is there, that means the organisms present, what they can do, that is their potential, what are they doing, and finally how do they interact with themselves and a biological environment. So, in terms of who is there, it is basically community composition that we get, but unlike the 16S ribosomal RNA gene which is only targeting the prokaryotic organism, in case of metagenomics it is beyond any boundary of prokaryote or eukaryote. It will give you all the taxonomic marker genes, 16S, 18S, even the RPO-B gene and many other single copy marker genes which are conventionally used to delineate the taxonomy of the organisms, both eukaryote, prokaryote, virus, everything. So, you will get the community composition beyond any taxonomic boundary. What they can do? If you remember earlier when we were trying to do this same answering the same question in NGS based methods or other methods, we are targeting genes, specific genes. But in this case, since it is relying on the total genomic pool, it will give you the metabolic potential through all genes. All genes present in the environment, that particular sample, the information about them will be in front of you, and since you have all the genes or information about all the genes, so you can actually build the pathways which are relevant for carbon metabolism, for nitrogen metabolism, for sulfur metabolism, for transport, for a disease development, for antibiotic resistant, for fatty acid synthesis, for carbon dioxide capture and sequestration into carbonate minerals, for any kind of pathway. Basically, you will be having opportunity to reconstruct all the pathways. Out of this, you will select that which pathway you want to emphasize more, provided of course, if you have sufficient good quality data with you. That is of course a point, and I will come to that point. What are they doing? That is again the fundamental thing, that the realized metabolic potential, that means the function that they are actually carrying out. So, the question number 2 was what they can do, that is the genes they have. Now, whether the genes are actually getting expressed or not, for that possibly we need to select RNA, and if we are taking the RNA out of the sample instead of a DNA, then the total RNA, that means it is the metatranscriptome. So, the metatranscriptomic sequencing in the same way would give us the realized metabolic potential, all the genes which are being expressed or are being function being carried out. So, and finally, how do they interact with themselves and a biological environment? Of course, for that we need to have the network and models, and we need to use multiple samples and multiple data sets. Now, I will give you one example where you can see that this is from the particular paper, that is on the metagenomic profiles of activated sludge from a full scale wastewater treatment plant. So, we can see that we have used this wastewater treatment plant sample, and when we perform the whole metagenome analysis, the scientists they perform this Guo et al, they perform the whole metagenome taxonomic profiling incorporating all the taxonomic information. You can see that it is again dominated by mostly the bacteria, and there are certain kind of eukarya and other organisms are there, and again it is mostly the proteobacteria, and at organismic like genus level these are the data, this is a corona chart we call it, and these are all the organisms and their relative abundance which is plotted over here. So, now you can compare this with since it is the wastewater treatment plant with different samples or under different treatment conditions, and then evaluate the composition. Okay. The next is the function, that is the very interesting thing. As I mentioned earlier, the function here is delineated not based on any particular target, any single gene, but it is rather based on all the genes which are present and all the genes which are detected. So, there are certain systems through which actually we annotate. So, here it is the cluster of orthologous groups annotation, COG annotation. So, through this annotation, when we try to annotate means we try to find out the meaning of those contigs, those genes that are present there, which type of reactions they carry out. So, generally there are certain systems that we have identified within the genomes or the genomic property based on that. So, we in this case these are the major categories that are highlighted, like the metabolism category, information storage and processing category, cellular processes and signaling category, and some other categories are there which are called poorly characterized. So, there might be other categories also, but for the simplicity the figure is containing only these 3 and 4, the poorly characterized one, because the group might have found some genes allocated to this. Now, within each category like or the system, the metabolism category, which is a major category, there are numbers of pathways. So, these are the pathways. So, these pathways are maybe some little higher level of pathway, which are, you can see, these are called subsystems. So, within a system of or category of metabolism, there are subcategories which are like secondary metabolism, nucleotide transport and metabolism, lipid transport and metabolism, inorganic ion transport, energy production and conversion, etc. So, now energy conversion and production, these has significance allocation of genes. So, these are the percent allocation. So, out of the total genes, those are detected. So, around 10% genes are allocated to energy production and conversion. Now, if we want to look into this particular pathway or this particular subcategory, that what are the actual pathways which are going on in this particular activated sludge or the wastewater treatment sample, which are facilitating the energy production, then we can do that, because we can find out that 10 pathways are going on, and each of these pathways are having maybe 10 step or 20 step or maybe 5 steps. So, all those genes are detected and their information are collected together, collated together, and they are put under the heading energy production and conversion. Similarly, transcription, replication and all things are done under the information storage and processing. Cellular signaling is also having many features. So, like that, eventually if we want to map the entire process happening in this particular sample, this is called the KEGG mapper, which can provide us that based on the genes which are detected and their relative abundance, what are the processes, metabolic processes are going on, and what is the overall nature, that we are talking about the systems level understanding. So, within the activated sludge or the wastewater sample, there may be millions of bacteria of several thousand species. So, rather than relying on the functions carried out by 1 or 2 or 3 or 4 species, if we can see that what is the total function carried out by this sample or the community of this particular wastewater system. So, this is the picture over here from this. If we resolve it, because it is not possible to resolve this over here, but if we can resolve it in a live online system of KEGG database, we will be able to see. For example, here if we go, we will be see that the citrate cycle or the TCA cycle is almost complete except these few steps. The initial few steps are missing, maybe this is bypassed by some other reaction, and somewhere here we have the glycolytic reactions and we have the other reactions of the energy metabolism and amino acid metabolism, which are also very active. So, we can readily understand that at system level how this community is behaving. Now, if we want to make use of this, like if we want to produce or look into this community, that whether we can produce certain useful molecule like some energy molecule, some lipid molecule, so that we can actually convert this process into a part of the circular economy or something like that, then we can actually fish out or we can actually look in detail of those genes, which will allow us to identify that whether these genes are there and how that can be helping us. So, some of the key points about the advantages of this whole metagenome. So, this is only a part of the whole set of data that can be generated. So, I just took one simple example, but there can be many more similar examples or maybe more sophisticated and complicated also examples are there. So, some of the key points which are actually to be identified as the major points of advantage are the fact that by examining the functional and metabolic attributes of the sequence genome, it is possible to have a more accurate identification of the microorganism present, because now we have not only 16S or not only 18S genes, but we have all the taxonomic marker genes. So, based on all the taxonomic marker genes, we can actually reach out to all the organisms which are present there. And also we have now the ability to predict the complete functional profile of the community, like the KEGG map that I have shown you, that is the prediction of the complete functional profile. Now, metagenomic approaches avoid the known biases associated with the PCR amplification, including the primer bias, where the primer miss certain taxa, or chimera formation, where artificial hybrid sequences may be created during PCR amplification and they will be thrown out or discarded during the subsequent quality check steps. So, bioinformatically extracting phylogenetic or functional marker genes, because here the whole data is with us, whole nucleotide and genomic data is with us, and it is only bioinformatically the reads are identified and extracted. So, that may offer a less biased method of surveying the microbial diversity than the metagenetic approaches or the target gene amplification approaches. However, it has one very severe kind of issue which we must discuss briefly, that it is not targeted. It is going to be for all the organisms in the sample. The number of sequences covering any given gene is usually far lower than could be obtained by metagenetic approach like the PCR based approach, because here we are not amplifying any specific gene, all the genes are available to us. So, suppose I am looking for a particular carbon fixation gene in my sample, but it may so happen that the carbon fixation gene abundance is naturally very low. So, when I have maybe 1 million reads and after 1 million reads are treated, I may found that my carbon fixation gene is not there. So, that possibly mean that truly the gene is not there, but it could also be possible that if I have like 10 million reads, then perhaps I can get that particular gene detected over there. So, meaning that far deeper sequencing is required. So, it is always better to have a deeper sequencing with respect to the whole metagenome sequencing, so that we do not miss out anything, any important, and we are able to actually construct the pathways, because for each pathway there are certain minimum genes required to complete the pathway. Any pathway would require a set of genes in order to complete the pathway, like from glucose to pyruvic acid, for example, in the glycolytic pathways we should have all the genes. If we do not have all the genes in our sample, then the pyruvic acid will not be produced. So, the minimum detection is desirable. So, at least 1 or 2 copies of the gene must be detected, so that we can say that this pathway is complete. Now, increasing the sequencing depth will give us more data, so that we can quantitatively compare this, that what is the quantitative abundance of a particular pathway with respect to another pathway. Second, and a minor issue, I will say that the financial and computational cost associated with acquiring and analyzing these data. For certain research groups and certain countries also, these are prohibitory, because this is naturally high. Nevertheless, the metagenomics holds great promise as a versatile technique for studying many aspects of microbial community ecology. So, metagenomics is not the end of the road. Following metagenomics, we have the metatranscriptomics, where we use the RNA. So, RNA instead of DNA, as I mentioned earlier. So, it is the same approach as we performed with the metagenomic approach, that is fragment the pieces or convert the RNA into DNA and go ahead with sequencing. But here if we take the RNA then and apply this technique, then we will be able to quantify which genes are actively expressed at a given time, and that is called the metatranscriptomics. And metatranscriptomics can provide a detailed picture of the functional and metabolic activity of the community, exactly what is happening in the community at the time of sampling. That is very important, because the community might have many pathways are complete, pathways are incomplete, many things are there, because if we do a DNA based study. But if we do a RNA based study, we will be able to find out very clearly that which pathways are operating under which condition, and also the organisms involved in that pathway also. So, and can be used to examine almost instantaneous changes to this in response to environmental perturbations, particularly in the reactors or in the tank where we treat different type of wastewater. So, metatranscriptomics is found to be or is actually working very well with that. Now, in addition to both these approaches that I mentioned, metagenomics I discussed in detail and metatranscriptomics, alternate omics approaches are also there, which are called metaproteomics, for example, considering all the proteins which are present, or the metabolomics, that is the considering all the metabolites which are present within the community. So, here I present some of the very important research papers which are found to be kind of path breaking research, I will say. So, like the first paper in the left corner is by Tyson et al. So, this Tyson et al. And the Venter, if you remember the Craig Venter, that who actually initiated and is considered to be the stalwarts of this sequencing, genome sequencing technology and the process. So, these two papers, the community structure and metabolism and the environmental genome shotgun sequencing of the Sargasso Sea, was the initial days paper. So, these papers showed the path that what could be possible, and subsequently many papers appeared. So, we are not going to have discussion on that, but I just want to show you one or two papers which might be important for you if you are interested. If you are interested on understanding how metagenomics tools are being used in microbial ecology research, this is a very recent paper, 2021 paper I believe, and there is also another paper, metagenomics microbial diversity through a scratched lens, and discussed certain other points. These two papers by Desai and Alberton, published in 2012, are also very useful because they raise certain critical issues and identified the history of development and what exactly metagenomics are able to provide from genomics to metagenomics, something like that. So, with this, these references are given because I found some of these papers are truly very useful for you to go through. For example, the last one, the streams of data from drops of water, is very comprehensive, and also the other papers which will be useful. So, there are numerous paper and other articles available that can be used. So, in conclusion, I will say the concept of metagenomics is briefly explained, and it is highlighted that this is the method through which we can actually gain access to the all the organisms taxonomic identity, of course, but also their functional potential. With this, I thank you.